right, welcome to your first online supporting lecture for In Bio 104, Biology of Plagues. This is Lecture 2, Part 2. We will talk about cell structure with a little history thrown in. The goals for this part of the lecture are to talk about what a cell is and what the bare minimum is that a cell must possess, some detail, and also to learn some detail on what is in prokaryotic and eukaryotic cells, talk about what parts are present and what those parts do, and we're going to talk about who first observed cells and how. Please be sure to take careful notes as if you were in class as you go through this lecture file. A cell can be defined as the most basic unit of which all organisms are comprised or made of. They are relatively tiny. I'll talk more about that in a moment. A microbe, by definition, is an organism that exists as a single cell. In contrast, multicellular life, such as the plants and animals, consist of organisms where one organism contains billions of cells. Cells can look very different from one another, but they all have common features. Examples of differing cell appearances are shown here in the variety of cells depicted in the figure. Bacterial cells are relatively simple. Bacterial cells are shown here. You saw cells like this, although perhaps the images weren't quite as clear. This one's been photo enhanced in your recent lab activity. You'll note that bacterial cells, although they're quite simple, are still metabolically complex and still able to achieve all of the functions required of cells. And as you'll see in a few moments, you'll learn that they have basically the same parts as those seen in eukaryotic cells. All of the other cells shown here are examples of eukaryotic cells. This is an example of a plant cell, or a group of plant cells, and the other three are examples of cells that are found within our own body. Skin cells, neural cells, or a neuron, and an egg cell, human egg cell. So what I like looking at when I look at or think about cells is I actually really enjoy looking at cells from the human body, such as the three shown here, and I really find it fascinating that even though these are all from the same organism, their appearance varies so drastically. You want to remember that each of them is specialized, which allows it to perform a particular body function. So that neural cell has very long axial processes, which can carry electrochemical signals, and these reach across the entire body through the nervous system, allowing us to control our bodies, respond to the environment, sense pain, feel pleasure, and so on. Our skin cells are there to protect us from the outer environment, to keep moisture in, to keep pathogens out, and they are relatively flat cells and slough off continually. So there's a high rate of turnover for the skin cell and a low rate of turnover for the very highly special neural cell. And the egg cell has its own particular design so that it can participate in reproduction. They're all very different looking, but they all have common features, as do all cells. That's what we're going to talk about now. Most cells are microscopic, meaning that they are smaller than the 200 micrometer. A micrometer symbol looks like this. It's a funky little U and then an M, and that's a micrometer. It's a metric unit of length, very, very tiny. There are a million micrometers in a meter. A meter is about the distance from one of your shoulders to the tip of one of your fingers. Anyway, most cells are microscopic, meaning they are smaller than the 200 micrometer width that the human eye can see, unaided by any sort of magnifying device. This image, the image shown in this figure, or in this slide, gives us a feeling of scale, taking us from the atomic level, over here on the left, to the tissue level, over here on the right. So cells fall in here. Okay? So here's a red blood cell. It's 6 microns, which equals 6 micrometers. A micron's just sort of like shorthand for micrometer wide. So you can't see that because it's smaller than 200 micrometers, the typical size something has to be for us to see it with the unaided eye. So the only way we can see those structures is if we magnify them, and that's what we use microscopes for. So similarly with bacteria, typical bacteria might be, anywhere from 5 to, 100 micrometers in length, going from the very small to the very large, and we can't see them with unaided eyes. This is true for most cells. I should add that the link takes you to a site where you can play around a little bit, and it's a really interesting interactive tutorial that will give you a bit of a sense of the scale of some of these really tiny objects and relate them to objects that you're maybe more familiar with. So let's consider now what the bare minimum is for cells. Like how simply can we talk about them and still be having a meaningful conversation? What do all cells have, and basically why do they have it? So all cells have cell membranes, which are often referred to as the cytoplasmic membrane. This bounds the inside and outside of a cell. In other words, it dictates what is inside and what is outside. And importantly, it controls what goes in and what goes out. So it's protective of the contents inside the cytoplasm. The cytoplasm is a watery fluid that fills the inside of a cell. Also in the cytoplasm are salts, proteins, sugars, and other molecules. Kind of like a soup. All cells also have chromosomes, and the chromosomes are made out of what is called DNA, or deoxyribonucleic acid, DNA. Okay? This is really important molecule, and it's important because it contains what we refer to as the genetic information, or the blueprints, that allow proteins to be made. And once you make proteins, proteins are a group of molecules that accomplish all of the work of a cell. That work is metabolism or energy processing, locomotion or moving around, transport, getting things in and out of the cell, and then a variety of other functions. That should be an R in there and a space. I apologize for that typo. So without proteins, you can't do cell work. So you can't reproduce, and you can't metabolize. So you're dead, basically, from a genetic perspective and a cellular perspective. So you need proteins. So the information to make the proteins is maintained in DNA. So all cells have to have both of these things. All cells also have common ways of dealing with energy, chemical energy, within the context of the cell. And the common so-called energy currency of cell is a chemical called adenosine triphosphate, or ATP. Okay? All cells have ATP. These are common to all living things and to all cells. That's the bare minimum. So, a long time ago, scientists came up with a list of qualities or characteristics that basically define life, or can be used to answer the question, is this or is that alive? Is it a living thing? Is it an organism? And these qualities are shown here. There's cellular organization, energy production / processing, I would say. Okay. Reproduction, irritability, or the ability to respond to the environment, and the ability to undergo growth and development, even at the level of a cell. Okay, so even a microbe can undergo growth and development, some sort of specialization here and there. Now in red I've put cellular organization because it's what pertains to this particular discussion that we're having. So all life consists of cells. Therefore, all life, if you look at its most basic building block, you see units that are organized in characteristic patterns, those of cells. They all have membranes encasing water-based solutions and cell structures. They all have DNA in the form of chromosomes containing genetic information, and they all have proteins and also structures called ribosomes, which I didn't mention a moment ago, that are used to convert genetic information into proteins. So in other words, it is the structure and the function of a cell It is cellular organization that actually allows for the qualities 2 through 5 to occur. Without cellular organization, 2 through 5 could not exist. I have discussed the idea of two fundamental cell types with you, but I want to focus really in this lecture more on how these cells are fundamentally similar instead of being different. We're going to take a tour through the composition of a prokaryotic cell next, and then we're just going to spend a moment or two looking at eukaryotic cells. But what I want you to hold in mind is I want you to be looking for the common elements. What do all cells do? What do all cells have? And then that will actually help you better understand these two different patterns, because you can ask, well, how do prokaryotes achieve the function known as maintaining genetic information? And how is that done in eukaryotes? How are they the same and how are they different? So I want you to be thinking about making a note that addresses that in your study notes. Let's look at the basics of a prokaryotic cell. So this could be any bacterial cell or any archaeal cell. Because our class focuses on infectious disease, let's talk about bacteria. So here's a simple bacterium shown here. We've looked at this cartoon a number of times already this semester. If we start from the outside in, let's have a category and let's call it appendages. These would be attachments to the cell's surface. I don't need that there. So the types of attachments to the cell surface that bacteria have are flagella, pili, and fimbriae. The flagellum is shown here. Okay, it's that long kind of brown structure. The colors are sort of meaningless in this context. They're just for making the cartoon pretty and easier to distinguish one part from another. Bacterial flagellum are very important in motility or movement. Other appendages that bacteria have are pili and fimbriae, and shown here on this particular figure, these little green structures are pili. Pili is plural, pilus is the singular. Okay, there are no fimbriae shown here, but fimbriae would be even smaller appendages. Bacteria use pili and fimbriae for a variety of reasons. They use them in some cases for certain forms of motility or movement, and they also use them for defense and offense. For example, pili can be used to secrete toxins. That would be an example of an offense function. Okay? A defense function might be the use of pili, actually more likely fimbriae, or pili for attachment to environmental surfaces. For example, attachment to the epithelial cells lining the human body. That would be a defense function from the point of view of the microbe, because it would allow the microbe to stay in place in an environment that provides it with food. All of these extracellular appendages are made out of protein, different types of protein, and some of them are known as pathogenicity factors. Okay, so a pathogenicity factor is an aspect of a microbe that allows it to be pathogenic, or in other words, able to cause infectious disease. Now, how could these things be pathogenicity factors? Well, let's think about secreting toxins. If you've got some aspect of your cell structure and you're a microbe, and it can be used to secrete a poison that harms your host, then that will make the host sick. And when the host becomes sick, usually there's some benefit to the microbe. So maybe some host cells degrade and the microbe gets more food. But in any case, it's something about the microbe that makes the host sick. So the microbe is using that function to be pathogenic. Okay, let's consider some other areas of prokaryotic cells. Moving in, let's think about the cell envelope. So not really shown on this, well actually it is shown in dark blue down here. We've got the word capsule. So many microbes, many bacteria in particular, make capsules. They make them out of carbohydrates or certain types of sugars. Carbohydrate and sugar you can think of kind of synonymously. And they make capsules in order to protect themselves. They provide protection from drying or desiccation, also attack from phagocytes. So phagocytes would be animal cells that can engulf and destroy an invading microbe. Okay, so if you've got a capsule, you might be able to protect yourself from that fate, and also from viruses. Viruses act as cellular parasites, and although there are many viruses that attack humans, there are also viruses that attack plants, and there are viruses that attack bacteria. A capsule can help to protect a bacterium against such attack. Capsules are also helpful in allowing bacterial cells to attach both to one another and to surfaces. So because of these features, capsules can be a pathogenicity factor. Remember, I defined a pathogenicity factor in the previous slide. So if you think back to that protective function, phagocytes, which I defined a moment ago, are really important parts of our immune system. If you've got a bacterium and it's invading you and it's got a capsule, and our immune phagocytes cannot engulf and destroy that bacterium, then they can't get rid of it from your body. And so that makes the microbe more able to multiply and infect you. And so that makes capsules a pathogenicity factor. Also part of what we think of as the cell wall, or of the cell envelope, is the cell wall, which I discussed in a previous lecture. I mentioned in that previous lecture that the cell walls of bacteria are made of peptidoglycan, a fusion macromolecule consisting of amino acids and carbohydrates, and that the role of the cell wall is primarily to protect cell from water stress, protect them from blowing up basically, or lysing from water stress. Also part of the cell envelope is the plasma membrane. The plasma membrane is made out of lipid. It's a special kind of lipid or fat called phospholipid, and it serves to control what goes in and out of the cell. It bounds the cell, and also in bacteria, it is the location of ATP synthesis, the energy currency for the cell. Now let's think about what's inside the cell. We'll say inside the cytoplasmic region. So all organisms have to have genetic material in the form of DNA that is in structures referred to as chromosomes. So in prokaryotic cell, we have a single chromosome, okay, one circular chromosome, okay, and it's shown here in green. If we pulled that out and stretched it apart, it would be one circular chromosome. That's what's typical for prokaryotic cells. Also in the cytoplasm, we have cell structures called ribosomes, and ribosomes are able to follow the directions in a molecule of referred to as RNA, which I'll discuss later in the course, and they use that RNA and actually are the structures responsible for making protein. So they do what we call protein synthesis. There are also a variety, this should be of, what we call inclusions. For example, different storage granules. One thing that many microbes store up is starch. Starch is a complex carbohydrate and it's a really good energy source. And so if there's excess food in the environment, many microbes will store it for later times when there isn't as much food. So they'll store it in the form of granules of insoluble or not dissolved starch. So there are other features inside the cytoplasmic region that we could discuss, but these are the basics, and I think if you can learn and remember these, you'll have a pretty good understanding of prokaryotic cells. So most of the things that I just spoke of a moment ago in reference to prokaryotic cells are listed in the central part of this slide. So here we see on the left a prokaryotic cell. It's a different cartoon, but it's the same basic design. And on the right, we have a eukaryotic cell. This is probably an animal cell because I'm not noticing a cell wall, but it could also be it could be a protozoa, although they also have cell walls. This is probably an animal cell. So let's think about what's similar here. We still have a plasma membrane, and it is bounding the cell, and it is indicated in blue on this figure. We still have that watery fluid referred to as cytoplasm filling up the inside of the cell. We still have DNA, except in the case of the eukaryotic cell, the DNA is in linear chromosomes, not a circle, but a line, and they occur in pairs. Okay? Something called being diploid. And all of the DNA is in the nucleus. Remember, U means true and carry means nucleus. And this purple structure here is the nucleus. Okay? That dark bit at the center of it is a particular region of the nucleus that we don't really need to get into in this case. Okay? So the nucleus is bounded by membranes, another feature or hallmark of eukaryotic cells, and the nucleus contains the chromosomes for the cell. We also have ribosomes, and the ribosomes in eukaryotic cells are found embedded in a layer of membrane called endoplasmic reticulum. It's radiating out around here, and it's continuing through here and here. Okay, so that's where the ER is, and it is holding many ribosomes, or holding the structures responsible for protein synthesis. It also contains a lot of proteins, and it plays a role in transport in the cell. Ribosomes are also found just distributed through the cytoplasm in eukaryotic cells, as is the case with prokaryotic cells. Now this cartoon implies that eukaryotic cells are typically bigger. We used to say that was true, but in the last 10 years we've learned that in the ocean there are many incredibly tiny eukaryotic cells, so we can't really make that generalization anymore, and so I won't really support that statement here. So using the same three categories that we applied to our discussion of prokaryotic cells, let's think about the basics of a eukaryotic cell a little bit more. Let's think about appendages. So do eukaryotic cells have these extracellular appendages? And the answer to that is that some do. So some have flagella. For example, spermatozoa use flagella in order to locomote up the reproductive tract of females. It's part of their function. They have a reproductive function. Their flagella are structured quite differently from bacterial flagella, but they serve a similar purpose. Some animal cells also have extracellular structures. For example, structures called cilia that are found on the epithelial or surface cells lining our reproductive and respiratory tracts. When women ovulate, it is cilia lining epithelial cells in the reproductive tract that move the egg down from the ovaries lower into the reproductive tract. Whenever you breathe, you're using respiratory cells or epithelia that are lining your respiratory system, and there are cilia that need to move deep in your respiratory system, and they're moving mucus up and out of your lungs. And in fact, it's the impairment of those cilia from cigarette smoke that is one of the reasons that smokers are much more susceptible to respiratory infections. So they do have extracellular appendages, and they are made out of protein, and they play typically a role in transport, or not transport, motility or movement. Do they have a cell envelope? Yes. Most of them have cell walls. The animal cells don't. but the algae, the fungi, the protozoa, and the plants all have cell walls. And do they have a plasma membrane? Yes, they do have a plasma membrane, and it's made very similarly to that found in a bacterium. Eukaryotic cells don't have capsules, however. That's a bacterial cell feature. Do they have a cytoplasmic region? Yes, they do, and in that cytoplasmic region, you have a watery fluid full of salts, sugars, proteins, and other small molecules. You also have chromosomes. They aren't found free-floating in the cytoplasm. They're found encased in a nucleus. You do have ribosomes. They are found in the membranes of the endoplasmic reticulum or free in the cytoplasm. And you also have additional structures that are known as organelles that are mainly structures wrapped in membrane. And an example of an organelle, it's very, very important, is the mitochondria. That's plural. It's mitochondrion for singular. It is an organelle that is responsible for ATP synthesis. Okay, something that the plasma membrane does in bacteria, but in eukaryotes it happens at the mitochondria. Okay, so let's try to sum up like similarities and differences, and this cartoon depicts one way of thinking about that. The center of this Venn diagram talks about similarities. So in all cells we have DNA as the genetic material. I would argue that it's in the form of chromosomes as well, or I would add to that. In all cells we have ribosomes responsible for protein synthesis, and we have similar basic metabolism. In other words, we have a way of making and using ATP, and those ways are pretty similar from one cell to the next. Okay? Other similarities I would say would be the cell membrane, okay, and the cytoplasm. Okay, all cells have those things. Something that prokaryotes have, they always contain or almost always contain cell walls. They're single-celled, and they don't have membrane-bound organelles. In other words, they don't have a nucleus, they don't have mitochondria. Eukaryotes do have a nucleus, so we could say they contain a nuclear envelope. That's the membrane for a nucleus. They often contain cell walls, so plants plus fungi plus algae plus protozoa. Okay. They include multicellular organisms, although they also include many microbes. And in general, they have a more complex and extensive arrangement of DNA. They have something called chromatin. So how have we learned so much about cells, and when did we begin this learning? In order to study cells, we had to have microscopes. As I said before, they're very, very tiny, and you can't study them without being able to see them. And in order to see them, you require a microscope. So who invented the microscope? So there are two answers to that, two scientists that were alive concurrently and doing their work concurrently. So I'll mention them both, although one is not really more important than the other. So the first I'll mention is a Dutch merchant. His name was Anton van Leeuwenhoek. And he lived in the Netherlands, or what's today's Netherlands, but formerly Holland. He was a merchant and a naturalist or amateur scientist. And in the era, or in the years 1673 to 1723, he wrote a series of letters to a society called the Royal Society of London. That society is still in existence today. I think it's the oldest natural science group still in existence today. And in those letters, Van Leeuwenhoek described small or tiny creatures that he referred to as animalcules. And he included a number of drawings and descriptions of them. He had examined samples of his own feces, scrapings of teeth, pond water, anything he could think of, he looked at it. And the way that he looked at it is that he devised a really sophisticated magnifying glass, and a reproduction of it is shown there on the left. The lens is in that little tiny area at the top, and he would put his sample on the head of essentially a pin, make adjustments, and study. And he probably devised these initially to help him look at merchandise such as fabric or material that he was shipping. so that he could do things like count the thread count in fabric, and then he thought of applying those same magnifying structures to the world around him. So that is documented as one of the earliest magnifying tools, but it wasn't a true compound microscope. That was invented by another fellow. And that other fellow's name was Robert Hooke. He was an English scientist and surveyor, sort of a Renaissance man of many trades. He was also a fellow in the Royal Society of London, so he was aware of van Leeuwenhoek's work, and he devised the first compound microscope. Okay, a compound microscope is one where you have both the objective and the eyepiece, and they are located at opposite ends of an adjustable tube. So you were working with compound microscopes in lab recently. You were using with binocular ones that magnify using each eye, and Hooke first came out with just the single tube style that you can still find in many high schools today. So just like van Leeuwenhoek, Hooke looked at a whole variety of samples, and when he was looking at a cork sample, type of wood sample, he noticed these regular patterns, and he drew them, and they're shown in this figure here, which was published in a book called Micrographia, very, very famous book. So the cork is worth mentioning because based on his investigations of cork, he's the one that came up, or is credited with coming up, the term cell. Okay? And that was the beginning of what we call cell theory, or the idea or the truth that all living things are composed of cells. So he wrote about this in Micrographia, and also in Micrographia he documented many microbes. For example, a fungus commonly found on bread. And so he may be the first person to officially document microbes, or it may have been van Leeuwenhoek. The record's not entirely clear on who came first. One thing that clouds that view is that Robert Hooke was very proficient in English, whereas van Leeuwenhoek spoke Dutch and was uncomfortable in English and a bit reclusive, so Hooke's work was better disseminated or shared with others. So with that, I will conclude this lecture, and I will see you in class soon.